Now, I've posed a very startling question here, which is, can a tumour, can a cancer cell have a baby? So let me explain what I'm asking here. What we already know is that at the very early stages of development, then cells are very flexible. You can remove cells from a developing embryo at the morula stage, and the cells that are being removed will just be replaced, and development will continue as normal. So you could remove half the cells at the 4-cell stage, and you'd still get a normal individual developing. Perhaps even more unexpectedly, you could push 2 morulas together, and they would form a double-sized aggregate. And if you implanted that into a mother, it would give rise to an individual which is also normal in size. In other words, it would regulate for the extra number of cells. Mostly these experiments are carried out in mice. As I say, it would be illegal to carry them out in humans. So you can create a multiple embryo from different sources. So what you can do is to take morulas from 2 different strains of mice, say a brown mouse and a white mouse. And you can push them together and allow them to develop inside a mother that had been prepared for pregnancy. And what will happen is that that will give rise to a mouse that's normal in size, but will be a patchwork. Some of the cells come from one set of parents and other cells come from another set of parents. So effectively, it's a patchwork mouse with 4 parents altogether. But you can do something even more dramatic, which is that you can take a morula from one mouse embryo, and you can take some tumour cells from another mouse embryo. And usually the tumour cells that are used here are developmental tumours called teratocarcinomas. And if you push the mouse cells together with the tumour cells, the teratocarcinoma cells, you get a patchwork combination. If you insert that combination into a mouse mother that's been prepared for pregnancy, in time she will give birth to a patchwork mouse, some of whose cells come from the parent mouse and some of whose cells come from the tumour. But the interesting thing is that the tumour cells are now behaving properly. They are not tumour cells anymore. Somehow they've been restored to normal behaviour by being combined with signals from a normal mouse. Now, let's imagine that half the cells come from the tumour and they are scattered throughout the entire body of the mouse. So some of them will be populating the ovaries or the testes in female and male mice respectively. So if we made 2 mice like that of different sexes and then bred them, some of their offspring will have tumours for mothers and fathers. And again, these mice are normal and they are not more liable to get cancer than any other mouse. So the developmental signals have, in this case, restored that developmental tumour to behaving normally. And that's a very striking observation. Now what I'd like to look at is the relationship between embryos and cancer. So let's look at some of the cell properties that underlie embryonic development. And the first is that cells can move about. So there's a lot more flexibility, cell migration taking place in a developing embryo than there is in the adult. So movement is a regular part of development. Also, embryo cells are dividing very rapidly. Embryos grow faster than the fastest tumours can grow. So there are high rates of cell division, especially when compared to the adult. And then cells begin as being relatively undifferentiated, and gradually they move towards differentiated states during the course of development. And then they'll interact with each other. So for instance, muscle cells will attract blood vessels towards themselves. That does not have to be specified by the genome in detail. You know that anywhere where a muscle begins to develop, blood vessels will grow towards it. And those tissue interactions are a valuable clue as well. By the time you get to be an adult, many of your cells are differentiated. Cell division has either ceased or is highly controlled. Cell movement is relatively rare. Now let's compare that with cancer cells. One of the big problems about cancer cells is that they can invade and spread, in other words, metastasize throughout the whole of the body. Another issue is that they divide very rapidly, much more rapidly than normal cells in the body. They also tend to de-differentiate. So the more de-differentiated they are, the more dangerous the cancer is. So if your cancer is biopsied and the cells are still well differentiated, the chances are that that's a more benign cancer than one where the cells have de-differentiated, where they're no longer showing signs of the parent tissue that they came from. We know that cancer cells can also attract blood vessels, just as muscles can in early embryonic development. And this is a serious problem. Once a tumour attracts blood vessels towards itself, it can then begin to grow rapidly, and cells can detach from the tumour and spread through the rest of the body in the blood system as a result. So what we can see is that in cancer, cell division and cell movement have escaped from normal control. So if we compare that with the things happening in the embryo, you can see that the cancers have in many ways reverted to the behaviour which is normal for embryos. The reason cells have the ability to move about, to invade other tissues, is because embryonic cells have those abilities and they move about in the body. So what this implies is that if we could understand the developmental mechanisms that lie behind embryonic cell behaviour, then that might well give us really valuable clues to understanding and dealing with cancer cells and their properties.